Good afternoon, good morning from my side. I'm very pleased to have the chance to present today and discuss with you a new concept of genetic testing, where we strongly believe that this opens a complete new era in modern medicine, and this is the whole genome sequencing technology. First of all, I would like to thank the organizers for giving me the chance to present some slides around that concept. And on the other side, I have to apologize. I had already booked my flights to India, but unfortunately, I was tested positive for COVID on Monday. And according to the German regulations, then we have to go in self-quarantine for a week. Therefore, I really regret not to be together with you during the wonderful conference. The message I would like to present to you today is the very simple one. And this is the strong belief that the whole genome sequencing will completely change our understanding regarding the early diagnosis of patients and implementing the concept of preventive medicine in the molecular medicine and the molecular healthcare system. This is mostly based on the fact that the pricing for whole genome sequencing is going down so massively. When I did my first genome about 6 or 7 years ago, we had to invest about $100,000. Maybe some of you have heard about the announcement from the company Illumina last week. They are planning to offer the sequencing of the genome with beginning next year for about $220, which covers the sequencing. So it goes exactly in the direction that the price goes down massively. And based on this assumption and the meanwhile already reality, we have started developing 2 years ago a concept that is exclusively focusing on the whole genome sequencing for the diagnosis and for the prevention of diseases in our patients. And allow me to quickly summarize what is so unique and what is so special around that. Number one, the genome is the first time in our life as geneticists having a technology that covers and combines all of the advantages of the genetic testing. Quantitative analysis, extremely precise qualitative analysis. So you can really position the genome as the one fits it all product. No need for further single gene confirmation, no need for technologies like MLPA, CGR array, qPCR, DHPLC, or whatever you have in mind. So it is a massive simplification of the entire process in getting the patients identified. Allow me to remind all of us regarding the Genomic England program, which is such a wonderful reference program. Genomic England started 6 years ago and has been in the meanwhile able, based on the free of charge offering to every cancer patient in UK and every patient being suspicious for a rare hereditary disease, to demonstrate that this strategy is not only reducing significantly the diagnostic odyssey and the diagnostic delay in our patients from more than 6 years to less than 6 months. It has also significantly reduced the costs for the healthcare system or for the payers from more than 22,000 British pounds to less than 2,000 British pounds. The genome covers not only the nuclear genome, it also for sure covers the mitochondrial genome. We have decided to make a 1000x coverage rate that gives you a perfect chance to quantify the differences you may see in the different mitochondriopathy to less than 5% regarding heteroplasmia. With these technical characterizations, you're reaching a diagnostic yield of about 77% in the mean and this is a mean over all of the close to 10,000 genomes we have done in the last 18 months, in areas with higher consanguinity cousin's marriage, it's even higher than 80%. And what is also so important, that we are starting developing an end-to-end solution. In the past, it was typically the situation that we have sent out from the laboratory perspective a report and had the belief that the job is done. That's totally wrong. In the meanwhile, we have a clear understanding that with sending out the first report, the job and the interaction with the clinician and with the patient should start. Why? Because there's nothing faster progressing in modern medicine than genetics. Every month, more than 1,500 different research papers that may change the interpretation of the genetic data. And therefore, we have decided to send out every quarter automatically an updated report to the clinician to enroll the most recent information, understanding of new genes, changing maybe of the interpretation of the individual variant. If you compare the genome with the exome, still there is the clear belief, oh, the exome is absolutely sufficient, what we are doing in the genetic testing. Please just pay attention to the significant increase of the diagnostic yield. In the exome, over all of the studies we were running, 46% compared to 77%. So it's close to double the diagnostic yield in our patients. We recently submitted a paper where we had the chance to test 1,500 patients from Pakistan. Why Pakistan? Because here we have the high level of consanguinity. You discussed this in the early morning. And with the exome negative cases, we have been able to add more than 40% additionally diagnosed patients based on the class 1 and class 2 variant we can take as the basis for the diagnostics. And the most important element is on the one side that the genome covers not only the intron and the regulatory stretches, it also covers as the only technology the complete compendium of the different structural variants you can imagine. And there is a further reason that argues for the superiority of the genome. And this is the fact that you are missing one important step that is mandatory in the exome. This is the so-called enrichment step. With the enrichment in the exome, you're generating artifacts. And here what we have done, carefully comparing commercially available enrichment kits. And you see on the right side in the first 4 lanes, in blue, in orange, in pink, and in gray, for 4 different enrichment kits, the difference is how they're enriching the different areas in the exon stretch for a gene that is given here as NAPB. And only with the genome in the lower lane in green, you see a very uniform and homogeneous coverage. And that's the reason why in a significant percentage, even in exon stretches, you are able to identify the causative variant only with the genome, and you have the risk to miss it with the exome. Allow me to quickly summarize the superiority. This is on the one side that exome negative patients demonstrating to 6 to 9% complex structural variants that are missed with the exome. 7 to 12% are demonstrating pathogenic, intronic, and regulatory stretches based variants missed by the exome because the exome is only covering the exonic structure. And last but not least, 6 to 10% of the exome negative cases will be shown to be positive for a causative pathogenic variant in the exon due to the higher uniformity of whole genome sequencing. And allow me just to show you some of the real life data. You may not all be experts in the analysis of the next generation sequencing data, But you can easily see in the lower lane where we are demonstrating the exome results, here shown for a gene named selenon, that on the left side, highlighted with a red box, there is an exon structure that was due to the enrichment problem that is sometimes inherent for the different technologies based on the exome. You have missed to cover this exon by the exome sequencing. Perfectly covered by the genome. That's the reason why for this patient, the initial report based on the exome was negative. The genome was perfectly able to cover it. There is a further element I would like to underline, and this is the importance of using in the future the right reference genome. In the past, and I would say still the majority of the laboratories are using Caucasian reference genome for the analysis of samples, for example, from India. This automatically ends up in 15 to 20% false positive or false negative results. We have to choose the right reference genome for the proper interpretation of the data. That the genome covers perfectly the different ethnicities, or even the different tribes, or even the different sub-ethnicities. I would like to show you with that slide here, and research data we have generated from Pakistan. In that case, based on more than 600 Pakistani people, where we have done the genome, and you can perfectly, with the right knowledge, here we did a so-called principal component analysis, substratify a population like the Pakistani population very precisely in the different areas where the patients are coming from, reflecting whether they are Pashtuni or wherever they are coming from. And even very closely located cities like Lahore and Islamabad, shown in green and in blue, you can perfectly discriminate just based on the genomic data. Therefore, the first step we are always doing, based on our more than 10,000 genomes we have already in our internal data bank, first checking where the individual is coming from and where do we have the closest alignment to use the proper reference genome. This has a direct impact on the quality of the diagnostic procedure. And allow me to show you just one slide underlining one specific patient we analyzed, again, real life data using our reference genome concept. On the left side, you see the patient without using the proper reference genome. You know, you start with the filtering. I will introduce this later on a little bit more into the details. Then you check the frequency in the healthy individual cohort for the specific variants. You have started on the left side with more than 320,000 variants. With a comparison to the frequency in the healthy individual cohort, you can reduce it to roughly 108,000. If you analyze the biological impact, about 1,000, then check for the HPO, human phenotype ontology match, you end up in 99 cases. And if you check for the proper mode of inheritance, pedigree is so important, you have ended up on the left side with 14 variants that has been seen as the potential explanation for the problem of the patient. Do it like it's been shown on the right side with the reference genome. You do exactly the same process. Again, you start with 320,000, but you end up only in 5 variants. So the difference of 5 to 14, you might argue, that's a small number. No, that's massively reducing the risk to come back with a false positive result from 14 to 5. This is significantly reducing the costs because computational time is in the meanwhile extremely important. It reduces also the costs for the medical doctors and for the clinicians to do the final step. With using the right reference genome, we have been also able to demonstrate that we can reduce the problem, which I personally see as a clinician mostly in the variant of unclear significance, the VUS, for more than 90%. Cohorts using the proper reference genome have 10% or 90% less variants of unclear significance variants that are reflecting the most significant burden to our clinical colleagues. Allow me just to introduce one case to you. This is a patient that has been introduced coming from Southeast Asia. If I remember it correctly, it was Indonesia, with a clinical diagnosis of epileptic encephalopathy. The symptoms that had been described by our clinical colleagues have been seizures, intellectual disability, global developmental delay, microcephaly, and motor delay. You see the pedigree, non-consanguineous family, and the whole exome was negative in this patient. What we are always doing during the diagnostic process, starting to understand what is the key message and the key knowledge we can extract from the clinical information. And you can describe it in this individual patient either as a neurometabolic disorder or neurodevelopmental disorder with epileptic encephalopathy. I guess this very clear first alignment in the understanding and the conclusion of the clinical diagnosis is really critical. Once you have done the genome sequencing, you end up roughly with about 4 million variants that reflects heterogeneity in our genome between all of us. You check, as I have shown you earlier, for the shared variants, for the frequency, for the biological impact, for the clinical similarity. And this is exactly what we have also done in this specific case I've shown to you. And we ended up with the identification of a variant being depicted here at the position 146C2A in a gene called NAPB. What is now the analytical process you should ideally follow? First, you check for sure international data banks. We have two different GNOMAD versions in the meanwhile, the GNOMAD version 2 and the GNOMAD version 3. 2 is the bigger one, but has less genomes. 3 is the smaller one, but has more genomic data. So if you are now checking for this NAPB variant at the position 146, we have identified this in the GNOMAD version 2 in one individual, resulting in a super low frequency that is being depicted here. In the GNOMAD version 3, it has not been described up to now. What you are doing now in the next step is you are taking all of the available prediction programs that have been depicted here, SIFT, Polyphene, Mutation Taster, and so on, and you check for the functional consequence you can predict for this specific variant. And without going too much on the details, you see on the right side, again boxed and highlighted with a red box, the prediction, which is in the overall majority either deleterious, abbreviated with a D, or highly pathogenic, abbreviated with an H. And here I summarize that. So consistently, we are ending up with a prediction that this variant is a damaging variant that causes a biological phenotype. The next step, we always ask ourselves, what do we know regarding the biology of an individual gene and the potential variant we have been able to identify? And for the NAPB gene, we are aware that this encodes a cofactor that is involved in the so-called SNARE complex, very well known for neuroscientists, which is depending on the synaptic vesicle fusion and the recycling. And therefore, you can predict if there is a dysfunction of NAPB that patients should demonstrate intellectual disability, neuromuscular disorders, seizures, and also neurodegenerative symptoms. Further argument is that the NAPB is more or less exclusively expressed in the brain tissue, which has been shown in the lower lane, highlighted with a red box. So these biological data again underline the potential biological consequence and therefore fit with our patient. Sometimes there are already transgenic mice as models existing. You are checking this as well. So here we had been able to demonstrate that, that mice being homozygous for a knockout allele demonstrate recurrent epileptic seizures. starting from the postnatal day 11 onwards. So again, clearly in line with our phenotype we have learned from the patient. If you are going to the PubMed and all of the, we call it mastermind, a system we have developed to extract more or less continuously on the fly all of the new papers, all of the publicly available data bank, we have been able to identify 10 entries. And I just highlighted in red the different clinical supports we could get from these 10 entries, something between rare epilepsy, dysfunction of synapse, also causing cerebellar symptoms, epileptic encephalopathy, early onset epileptic encephalopathy, and so on. Also from the scientific papers, there is a good fit explaining and making it extremely likely that this individual variant I've been shown to you is really explaining the phenotype in our patient. If we sum up the evidences we have been able to collect for this individual case, we can clearly describe the model of disease is based on a disorder of synaptic vesicle fusion machinery exclusively affecting the brain tissue. Autism recessive, we have the mice model, the transgenic mice model, and we have a perfect clinical match with what has been published in the literature up to now as causing developmental and epileptic encephalopathy. The only piece that was missing in this specific case was that we didn't have access to a 3D protein structure that is underlying the functional positioning of the described variant, and having ideally also a functional assay that allows us to predict the protein dysfunction. However, beside that, this is a nice example how on the one side, you should go through all of the different layers of evidence once you have identified the variant to make it really a proof that you are able to identify the reason for the biological problem in your individual patient. I would like to go with you to a second case. We always appreciate getting as precise as possible clinical characterization of the patient. And do me one favor. We are all aware the doctors are burdened by the time pressure they have. It's difficult for them sometimes also if they are not clinicians having a huge experience in genetics, or pediatricians, for example, being very well educated in genetics, that sometimes for the doctors, it's not so easy to extract the right information, what might give a hint towards a direct interpretation and a proper interpretation of the genetically generated data. Therefore, what we always appreciate, if we are just getting the raw data, the medical letter, the handwriting for the doctor, and then extracting based on our mastermind technology, the information by ourselves, because this has the advantage that we might not run into a bias. So allow me to say, as a neurologist, sometimes neurologists have problems to understand the involvement of the heart or the bone or the liver or the lung, and therefore they are clearly focusing on the brain and the peripheral nerve. But it might be of relevance to understand that also the heart might be involved. So let me present this case to you. This is a 6-year-old boy from consanguineous parents from India, no specific disease in the family history. And here we had the pleasure to receive a detailed description without making any suggestion regarding genetics from our clinical colleague. And our system highlights what is with highest likelihood relevant for the understanding of the clinical phenotype. And I've been able to publish some years ago a paper, even though it was a small paper, but I would say one of the most important, most frequently referenced papers, because that paper was demonstrating a simple result. The more clinical HPO terms our clinical colleagues are presenting to the laboratory, the more likely the patient will be properly diagnosed. One HPO term based on exome data in that time was resulting in 29% of a diagnostic rate. The same patient with more than 15 HPO terms increased the diagnostic rate to more than 42%. So without investing a further dollar or euro, the doctors, the clinical colleagues have been able to significantly increase the likelihood of a proper diagnosis, having done the exam in that time, just by delivering more detailed clinical characterization of the patients. Therefore, do me the favor, whoever is your partner from the laboratory side, give the lab the chance to understand the clinical phenotype. And the lab for sure has to properly extract this. So for this patient, an exome sequencing was done July last year. It was negative by a third-party laboratory. We did in January the genome, and it was initially negative. I mentioned to you that we are sending out every quarter automatically an updated report. So we reanalyzed the case end of March, and suddenly it was popping up as being positive. And the interesting question is, what was the explanation? And the explanation has been shown here. For covering large deletions, insertions, recombinative events, you need a perfectly optimized pipeline based on the genomic data you are generating. And we are working intensively on that. Recently, we submitted a further paper demonstrating that we have now reached a diagnostic sensitivity and specificity of more than 99.85% for the identification of any form of a structural variant. And for this case, we used our structural variant analysis. We call it the Manta platform, again in March. And with that optimized platform, we identified a large deletion that has been depicted here for an exon 7 in a gene that is called ZNFX1 that causes a mitochondria-based pathophysiology resulting in an immunodeficiency type 91 and hyperinflammation syndrome. This is perfectly fitting with the clinical phenotype. Some of you who went through the slide before have realized that the most important clinical spectrum the patient is describing is an immunodeficiency syndrome shown and based on recurrent infections. So this gene initiates immune responses against RNA virus infection. If there is a genetic defect, it has been associated with immunodeficiency, and typically the patients are demonstrating different features that might include fever, thrombocytopenia, renal or hepatic dysfunction, recurrent infections, and if the brain is involved, also seizures. Sometimes we also see intermittent monocytosis and hemophagocytic lymphohistiocytosis-like phenotype, which was also the case in our patient. And just to make it more understandable and easier, you see very nicely in the upper lane exactly the resulting figure of the whole genome sequencing of this individual patient. And you see very nicely this deletion that had been now identified for this patient. This is a nice example how important it is to continuously work on the reanalysis of all of the cases, because this implements more and more the continuous improvement of the bioinformatic pipeline, which is in the meanwhile the most important contribution. And allow me to summarize the actual situation of the whole genome sequencing. The whole genome itself, from the technological basis for the generation of the data, that's nicely standardized. This is continuously improving regarding reduction of the processing time. But what still needs a significant improvement from the majority of the commercially available pipelines is the improvement of the bioinformatic-based interpretation of the data sets, plus the concept to continuously reanalyze the cases, and with that implementing the newest understanding of the improvement of the pipeline, plus new papers, new research data, progress of the data banks that facilitates the proper diagnosis in these patients. For a patient like this I've shown to you, it is so important that the patients are early identified, because what is very well known is the stem cell transplantation in these patients is getting more and more the golden standard. Alternatively, also immunoglobulin treatment might be an option. So we are realizing that more and more genetic diseases we are identifying have a direct therapeutic impact to our patients. With that, I would like to come to my end and would like to summarize. Already per today, the whole genome sequencing is getting the golden standard. Take the Genomic England program as a very nicely published and understandable reference program in the meanwhile. It reduces significantly the diagnostic delay to the benefit of our patients. It reduces significantly the costs that have to be spent to get a patient identified. And with the concept of the continuously reanalysis of the data set and send out such a report to our clinical partners, you're continuously increasing further on the diagnostic yield. I mentioned at the beginning that with the first and initial doing of the genome, we are already per today ending up in the mean of 77% of a diagnostic rate. With the first 12 months having done 4 further quarterly updated reports for more than 5,500 patients, we have been able to demonstrate that we can add further 6.5% of patients getting diagnosed based on the improvement of the data banks, based on the improvement of the description of the clinical phenotype, and based on the continuous improvement of the bioinformatic pipeline. And the last patient I have shown to you is a nice example how important it is not to believe that we're sending out a report, the job is done, but having the concept that the long-lasting interaction with the clinicians, with the clinical partners, and maybe also laboratory partners, we are continuously offering to the patients a further improvement and a further increase of the quality of the data. Coming to the end, I would like to announce you, some of you may know me since the last 15 years, having been with a former company very active in India. I strongly believe, and this is the reason why I founded the new company Arcensus, that with a genome exclusively concept, especially for a country like India, that for sure appreciates the best quality for the lowest price, we will be the perfect partner for all of you. And therefore, we are very happy that we have been able to identify in the meanwhile already a local partner, which is Solly Healthcare. Some representatives are already present with you. They are happy to answer all of your questions. Thank you very much. Unfortunately, I can't hear you.
Thank you, Professor Arndt, for such an informative talk. Now I would request Dr. Ashok sir to kindly give your valuable comments, and if you have any questions, please.
Thank you very much, Arndt. It's nice to see you, although it would have been nicer to have seen you in person. That's and I'm sure all of us enjoyed your talk thoroughly. It's really opened up some new thought processes, and particularly your emphasis on the taking it to the bedside and making sure that the result got to the family and clinical action was taken. It's not simply a question of posting out a result. That's the worst thing that could happen, actually. I really enjoyed it, and I'm sure everyone else did. Are there perhaps any questions?
Ashok, a pleasure to see you again and hear you again. Thank you.
Thank you once again, Professor Arndt. Now I would request the chairpersons for the second session, Dr.
Thank you
Ashok
very much.
Velodi, sir, and Dr. Manta, to kindly come on the stage so that we can proceed for the next